Now we're going to see the conjugation in the DNA bacterial transfer. So, conjugation is between a positive cell and a F negative cell. These positive cells could be F positive or HF positive. So this depends on how is the material DNA that is going to be passed from the cell to the cell. This is the bacteria has, let's talk first about the F positive cell. Has its genome and a plasmid. And this plasmid is going to be transferred to the cell that just has the material DNA. It could be different kinds of cells. So this plasmid is going to be transferred to this cell. The plasmid, here is the plasmid in B, has a part that is called the ority. Here it would break and would leave just one output, the two strands. Would break, and so one of these strands, just one, this strand is going to be passed to this cell, and then when this cell has this new strand, and this cell has now just one strand of DNA, each cell is going to transcribe the double-stranded DNA. And so now both cells would be F positive. This will become F positive, and this will stay F positive. And other thing is, in the plasmid that is going to be transferred, we have a part called the traoperon. This traoperon or region contains the proteins required for transfer of the plasmid. So movement of the plasmid. Next, proteins that allow the meeting between these two cells. That is this cell expresses a pili. This pili or pili meets with this cell and bring them together, and now they can pass through here. This plasmid is going to pass, well, one single strand is going to pass to the other cell. So we have the pili. That pilus, sex pili is genetically represented here. It is codified here, and all the protein needs for the plasmid to pass through them. But this part is the last part of the material DNA that is going to go there. So first we have a break in the ority, so all this strand goes through here, and at the end the tra is going to pass here. So this is the DNA passing through. This part is the traoperon, is the last one to be passed. In AF plus and F minus meeting, that do not produce any change. That's that the traoperon, the last part that we transfer. But in the, well, this was for F positive and F negative. Now for the HF positive and F negative, here is important because here we have the material DNA here, genetic material, and instead of having a plasmid that we are going to transfer to that cell, we have here this DNA is going to be transferred to that cell. But in order to pass this, we should pass all the genetic material, because here is the ority, and here, let's see, here is the traoperon. But the transfer began from, well, let's say that here is ority. The transfer began from here all the way down, all the way down until here. So should we transfer all this material in order to finally transfer the tra-operon. So first, yes, DRT, we have a break, and we are going to pass from this double-stranded DNA and here's the P. Just one single-stranded DNA, we begin with this green part, so would be the most advanced here, and then this black, and at the end, the tra-operon. The tra-operon that codifies for the pili and for the genes codifying proteins required for the transfer, for this conjugation, for the transfer of DNA from the cell to that cell. The problem is that this is a very big genetic material because it's all the DNA of the bacteria. It is not just a plasmid like here. So it's very difficult that all this passes through here because the meeting time is very short. So usually we just pass something like this. And so yes, there are some genes that are passing from here to here. That this is now in the cell. Yes, this green part that is the important that we are transferring can codify for some resistance to antibiotics, capsules, exotoxins, etc., and that confer advantage to this bacteria. But because we do not transfer the bacteria do not transfer the tra-operon, then this cell remains F negative because this cell doesn't have the tra-operon, so cannot initiate another meeting with other F negative cell because this cell cannot do the pili because doesn't have the tra-operon. So for that, this is F negative and this remains HF positive. That's the difference between HF positive and F positive cells. That because here is a plasmid can transfer all, and so this becomes F plus. But because here the DNA that is going to be passed is inserted into the genetic material of the bacteria, then just one part of the DNA can be passed through here, and the tra-operon is the last to be transferred. So the tra-operon never arrives to the new bacteria, and so the bacteria remains F negative. But this DNA confer advantage to the bacteria and is also going to be inserted into the DNA. So here is the bacteria, here is the genetic material of the bacteria, and this part is this part. And this is going to be inserted here. So let's do room here and here. So now it's part of the genetic material of the bacteria and confers advantage, but could not initiate another meeting, could not pass DNA from the bacteria to other because this remains F negative and cannot put the pili. Next, this insertion of this DNA to this chromosomal material of the bacteria is made by homologous recombination, as we talked in the previous video. So all the going to talk about bacterial genetics needs a homologous recombination. Homologous recombination except the F plus, F minus. These do not need recombination because here the plasmid remains here and will pass through this cell, passes and remains here in a plasmid. It is not going to be inserted, or they are not going to exchange material between the plasmid and the chromosome. So transformation, transduction, and conjugation all need homologous recombination except the conjugation between a F plus and F minus cell. There's also a thing that is this part of the DNA that is going to be transferred, but we could not transfer all of that DNA. If this DNA breaks, this part broken of the DNA can also go to other cell. That could be other way of doing it. Theoretically, also could be the trauperon, but that's very rare. The usual stuff is as I say.